Multiplex ligation-dependent probe amplification, or MLPA, is a genomic technique used to detect copy number variants in the genome, like deletions and duplications, in addition to insertions. MLPA is known for being cost-effective and allows for the simultaneous analysis of multiple DNA samples, a process known as multiplexing. MLPA is made up of 5 main steps: probe design, hybridization, ligation, PCR amplification, and data analysis. Let's jump into each of those. Probe design is the first step in MLPA. Here, 2 probes are designed to recognize a specific DNA sequence. The 2 probes are known as the left hybridization sequence, or LHS, and the right hybridization sequence, or RHS. The LHS comprises a specific hybridization sequence and a PCR primer sequence, and the RHS comprises a specific hybridization sequence, a PCR primer sequence, and a probe-specific STUFR sequence. The STUFR sequence acts as a unique barcode that allows for the identification of different biological samples, which allows for multiplexing within an MLPA reaction. The PCR primer sequences allow for downstream PCR amplification. Designing these probes increases the sensitivity and specificity of this technique through DNA-specific annealing, as they have complementarity to the targeted DNA sequence. The next step, hybridization, starts with denaturing the target DNA into single-stranded DNA at a high temperature. The 2 probes that we designed will be annealed next to each other on the single-stranded DNA, annealing to the targeted complementary DNA. Ligation, the third step, happens next as a ligating enzyme, known as a ligase, is added to the hybridized DNA. This effectively glues the 2 probes together, creating a single consecutive strand of DNA containing both PCR primers, both hybridization sequences, and the STUFR sequence. If there isn't complementarity between the probes and the target DNA, the ligase will not glue the probes together. Following ligation, PCR amplification is the next step, where PCR occurs in the traditional manner using the primer sequences included in the probe design. The double-stranded probe DNA strands are denatured before the PCR primers anneal to the primer sequences in our probes. Amplification of the probes takes place as DNA polymerase binds to the primer sequences and builds the complementary DNA sequence. This process runs for the desired amount of cycles to reach sufficient amplicon numbers for further data analysis. Finally, the PCR products are quantified using capillary electrophoresis as part of the data analysis step. Capillary electrophoresis separates samples by size, allowing DNA samples with varying STUFR sequence lengths to be separated. This is the key to the multiplexing aspect of MLPA, so that multiple samples can be run simultaneously while being uniquely identifiable. Let's check out what we've learned so far. Can you probe out the answers to amplify your understanding? Pause the video now to try it out. What does MLPA primarily detect? MLPA primarily detects copy number variants. Using the specific hybridization sequences, MLPA can bind to its target DNA and undergo amplification only if there is complementarity. In what step is the specificity of the probes to the target DNA most important? Probe specificity to the target DNA is most important in the hybridization step. Without specificity, the probes could either bind nonspecifically across the genome or not bind at all. What technique is used to quantify the amplified MLPA output? Capillary electrophoresis is used to quantify the amplified MLPA output. This helps us visualize the size and amount of the sample for quantification. MLPA is commonly used in both clinical and research settings to diagnose different genetic disorders, such as hereditary cancers or spinal muscular atrophy. Since the severity of SMA depends on how many copies of the SMN1 gene there are, MLPA can help determine the exact number of copies of SMN1 and help with disease diagnosis and treatment. In this electropherogram, we can see the peak heights for several samples in the region of gene X. The control samples all have peaks around 100 arbitrary units, while the peak for this patient sample is at 150 arbitrary units. Each control sample should include 2 copies of gene X, which means that our patient sample likely has a copy number gain in this region. But why might we choose MLPA over other known techniques? MLPA has many advantages, including the fact that it has high specificity and sensitivity for detecting variants like CNVs. It's less expensive than other techniques for CNV detection, such as a microarray, and it provides us with the capability to multiplex samples. MLPA also has a medium throughput or bandwidth, which is higher than popular techniques like qPCR. However, MLPA is not the perfect tool. While it can be designed to detect them, MLPA is not ideal for SNVs or small indels, as the variants must be known beforehand in order to design specific probes for them. It also falls short of the mark in sequencing the genome. We don't gain any sequence information, which also means that we don't know where the variant is in the genome or what orientation it's in. It's time to ligate your knowledge again with some real-world application. Take a second to try out this question. A patient is suspected of having a genetic disorder associated with a deletion in gene X. After performing MLPA, the electropherogram shows the following results. Control samples have a peak height of 200 arbitrary units for the region corresponding to gene X. The patient sample has a peak height of 100 AU for the same region in gene X. Based on the electropherogram, what is the copy number status of gene X in this patient? The patient likely has a heterozygous deletion of gene X. Since the electropherogram shows a peak height of 100 AU for the patient sample, while the control samples show a peak height of 200 AU, we can see that only half the typical amount of gene X is observed, suggesting a loss of 1 copy of the gene. Aside from the previously mentioned applications, MLPA can also be used to analyze methylation patterns in the genome using a derivative known as Methylation-specific MLPA or MS-MLPA. The main steps of MS-MLPA are almost identical to typical MLPA, but with a few key differences. In MS-MLPA probe design, the target sequence would be a region in the genome that is typically affected by methylation in disease. The rest of the probe design remains the same, with the LHS and RHS including the same components as before. Hybridization and ligation both take place as previously described, but a new step is introduced right after. This step, known as HAHA1 digestion, uses a restriction enzyme called HAHA1 to cut both the annealed probes and the target DNA at GC-GC target sequences. HAHA1 can only cut through DNA if it is unmethylated, so unmethylated regions of DNA are cut and cannot be amplified further down the line. However, if the target sequence is methylated, HAHA1 will not be able to cut through the DNA, and the DNA can then undergo PCR amplification. Following HAHA1 digestion, any methylated regions of the DNA that remain bound to the probes undergo PCR amplification. This exponentially amplifies these methylated regions and allows us to analyze the methylation patterns of different regions in the genome. These fragments are then visualized using the same capillary electrophoresis step as before, providing insights into both copy number and methylation status of genomic regions. MS-MLPA provides us with the capability to look at both the methylation status of the genome as well as the copy number. A typical scenario one might see this being used in is testing for Prader-Willi syndrome. In Prader-Willi syndrome, the 15q11 to 13 region is maternally silenced. This is called imprinting, and it happens when the alleles inherited from the mother in the PWS genes are methylated. In a healthy individual, the maternal allele is methylated and silenced, and the paternal allele maintains gene expression. However, in PWS, the maternal allele is silenced and the paternal allele is missing. The paternal allele might have been deleted, or something called uniparental disomy might have occurred, where 2 copies of the maternal allele were inherited instead of 1 from each parent. MS-MLPA can help us check to see if this region shows typical methylation and copy number status. We can quantify the amount of each gene and the amount of methylation to help inform us if someone is showing typical methylation patterns or if it might be indicative of PWS. As the future of genomic technologies grows and improves, MLPA will also continue to evolve and will be able to integrate with things like next-generation sequencing to further improve its accuracy and precision and open itself up to even more applications. As it stands, it remains the golden standard for detecting copy number variants and methylation changes in the genome and is a vital tool in both the clinic and research for its reliability and utility.